a protein coding gene. Okay. Not all open reading frames code for proteins. So the problem is to identify those open reading frames that code for proteins. So here are the 3 steps in identification of genes. The first step is to find features, statistical features that can discriminate between genes and non-coding regions, non-coding sequences. Okay. So identify features, statistical features that can help discriminate between protein coding sequences and non-coding sequences. This is the first step. Once you have identified those features, then build a model for each of these sequence types. One type is protein coding, the other type is non-coding, as an example. Okay. So once you have built the models, the third step is to integrate these models into a pattern recognition algorithm that can help discriminate protein coding genes from non-coding sequences. Clear? The 3 steps, and we will go through all these steps in a moment. Okay, so from 1986, Mark Borodovsky, and that time in undivided USSR, he was a scientist in a university in Moscow, and he was examining the sequences of E. coli, coding sequences of E. coli, and there were only handful of such sequences, and this is the pattern that he observed. So frame 1 indicates codon position 1. You understand? So if I write a sequence, and you did actually homework also, and in the first homework I told you that the first nucleotide is located in codon position 1. Then you know that the second nucleotide is in codon position 2, the third nucleotide is in codon position 3, and so on. So that would be first codon and then second codon and so on. Okay. So frame 1 here means codon position 1. So just look at codon position 1 throughout the sequence. Okay. Just look at position 1 only. So 1, 2, 3, 1, 2, 3, 1, 2, 3, it would be like this, right? If there are n number of codons, you have to just look at position 1 and the nucleotides in this position across the sequence. Okay. So this is what they did, and codon position 1 here indicates frame 1, codon position 2, frame 2, position 3, frame 3. Okay. And they observed that, Borodovsky and colleagues, and they observed that there is a non-uniform pattern with respect to the codon positions. So you can see the probability distribution of these 4 nucleotides at position 1 differs from the probability distribution at position 2 and position 3. Okay. So you can observe more frequency of A or G at position 1 compared to position 3, for example. So more occurrences of nucleotide A or G at position 1 and C or T at position 3, or so on. So they started thinking about the biological regions for these patterns, and there could be manifold regions. For example, and this is what they thought. They investigated and they came up with some regions, and one of the regions was that coding sequences lack stop codons, right? Coding sequences should only have codons that code for amino acids. And what are the stop codons? TAA, so TAA, TAG, TGA, so they always start with T, and at codon position 1, at position 1, right? So if you look at position 1, the frequency of T is much lower. So maybe that is contributing to that, many different regions. And then they also observed that, and this I think actually was known much before, that there is an abundance of RNY codons in coding sequences where R is purine, okay, purine, and purine is A or G. Y is pyrimidine, and pyrimidine is C or T. Okay, and this is position 1, this is position 2, this is position 3. So at position 1, there is an abundance of A or G, okay, because of the abundance of R and Y codons in the coding sequences. So which is reflected here. The probability of A or G, add them up, is over 60%, right, at position 1. And the probability of C or T at position 3 is, so let's look at this, is about 55% or so, okay. So because of this region, the abundance of R and Y codons, you can see, you can correspond actually. So it could be the potential regions and why you are observing this pattern. Also, there could be manifold regions, as I said, it's really complex. You know the degeneracy of the genetic code. So some of the codons could be preferred for an amino acid. For example, GGU could be used 90% of the time, and rest GGC, GGA, GGG could be used 10% of the time for encoding glycine. Okay, so there would be more use because GGUs are used more frequently. So at the third position, you will have more U's or T's, right, in the coding sequence. So yeah, all these together contribute to the differential pattern that is reflected in the coding sequences. Okay. On the other hand, if you consider non-coding open reading frames, and you can put the codon, even though they do not code for proteins, you can still put the positions. So first triplet of nucleotides 1 to 3, position 1 to 3, next 1 to 3, and so on. Okay, and then obtain the probability distribution for the 3 frames. And it was done, and it was observed that the probability distributions at the 3 positions, they are more and less similar. Okay, similar and same as the probability distribution for the entire when the positions are ignored. Okay, so which gives a hint that there is a pattern that discriminates protein coding sequences from non-coding sequences. Okay, and these are which kind of patterns? Position specific nucleotide compositional pattern. Okay, position specific, frame specific, nucleotide compositional pattern. Okay, so people started thinking about all types of features that they can consider for discriminating protein coding genes from non-coding genes, and one of those features was this. So this led to the development of algorithms. Many of those were based on Markov chain models. Markov chain models have been used frequently in the field of omics because of the reason I told you, that these are pluralistic models, and because of the ease of using these models for analysis of biopolymers, which are sequences of symbols, right? Those symbols could be the components that make these biopolymers. So because of these reasons, Markov chain models were adapted or adopted and were used in modeling of biological data. So let's look at what a Markov chain is, as mentioned on the slide. So Markov chains characterize stochastic processes. Stochastic means random processes. Random processes that incorporate a finite amount of memory. So it's not completely random. It is not memoryless. It does incorporate some amount of memory. Okay, good. A Markov chain model is used to describe systems that are governed by random processes with some amount of memory. Okay, So systems that can be described by such models that incorporate some amount of memory. Okay, so DNA and protein sequences may not follow Markov chain models. Okay, but if you consider these models, and because of the correlations that are there in these sequences, and correlations can arise for various reasons, these models can capture those correlations and can discriminate. So far the task of discrimination is concerned, these models can do it, even though DNA and protein sequences may not be following exactly the Markov chain models. Okay, so the goal is to use the models to capture the patterns that can help in addressing a problem. And that Markov chain model did, and that's why it became so popular. Okay, so let's consider DNA sequence, which is CGG, AT, and so on, and last is GC. Okay, if each base occurs independent of the other, which means there is no memory, they occur independent of the other, they do not have memory of the previous base. So then the model is of order 0. The system has a memory 0. Okay, okay, very good. Finite memory, which means that each base occurs dependent on some previous bases. Okay, so there is some memory of the previous bases. So if the occurrence of a base depends on just previous base, for example, if occurrence of T depends on the just previous base A, there is a memory of how many nucleotides, how many bases? One base only. Yeah, sometimes I will say base or nucleotide means the same. Okay, so means memory 1 and Markov model of order 1. Okay, if the occurrence of a base depends on 2 previous bases, then the memory is 2, and this is Markov chain model of order 2, and so on. Okay. Okay, so very good. If you understand this, then I can move ahead and I can let you know that Markov chain models were used for describing non-coding sequences and also protein coding sequences. So in the non-coding sequences, as I just showed you and told you, that the probability distribution of nucleotides across the 3 frames, they are more and less similar. Okay, so which means that you don't have to consider the codon positions. Of course, non-coding sequences do not code for proteins, so you can ignore the positions, and what you can do is to make a homogeneous Markov chain model of non-coding sequences. Homogeneous means not considering the positions. Okay, homogeneous with respect to the positions. So homogeneous Markov chain model of non-coding sequences. So frame independent or codon position independent. So let's consider this sequence. And if I ask you to compute the probability of this sequence for Markov chain model of order 0. So probability of this sequence given Markov chain model of order 0. Okay. So order 0 means that each of these nucleotides occur independent of each other. And you might have come across probability of A and B when A and B are independent of each other. Okay, so probability of A and B when A and B occur independent of each other is probability of A multiplied by probability of B. Okay, the multiplication rule for probability. Okay, so if each of these bases occur independent of each other, then the probability of S is probability of C. Yeah, same thing, apply this. Probability of C multiplied by probability of G multiplied by probability of G, and so on. The last is probability of G. Okay, Markov chain model of order 1. Okay, so the occurrence of a base depends on the just previous base. So, okay, so if I am here, okay, there is no previous base, right? So, okay, so I am going to, so I can start in C and then I can move to G. So, if I am here G, the occurrence of G depends on C, okay. So, I will start in C, so probability of C, so this is the initial probability of starting in C and then making a transition to G, and which is indicated by probability of G given C, okay. And then G, so probability of G given G, and last and so on, and last is probability of G given G, okay. So, starting in C, so you can sometimes say starting in C, making transition to G, then making transition to G and so on, finally in the end making transition from G to G. Clear? Okay. Markov chain model of order 2. Order 2 means that dependence is on two previous bases. Okay. Okay, so two previous bases. So if I am here, no previous base. If I am here, only one previous base. So make a transition to this. Now you have two previous bases. Okay. So the probability of starting in CG, okay, probability of starting in CG, dinucleotide CG, and then making a transition to G. So probability of starting in CG and then making a transition to G, which would be indicated by probability of G given CG, and then probability of A given GG, and finally, and so on, and finally probability of G given TG. Okay, so you can thus model a non-coding sequence using a homogeneous Markov chain model. Okay. Okay, so okay, good. Yeah, some details I am skipping because of the time constraint we have. But I would like to see that you are still appreciating these mathematical concepts that were used for describing biological sequences. So, protein coding sequences, they are made up of codons. And when there are codons, then you can visualize the codon positions, 3 codon positions, okay. So, which would indicate, and we saw that the probability distributions at 3 codon positions, they differ from each other, okay. Which means that there are some biological reasons why they differ, and you would want to capture this information, this biological information within the model framework. And that model framework is an inhomogeneous Markov chain model. Inhomogeneous means codon position specific or frame specific Markov chain model. Okay. Okay, so let's consider protein coding sequence modeling using inhomogeneous Markov chain model. So again, the sequence is the same, CGG, ATGG. Okay. What you don't know whether C is situated in codon position 1 or 2 or 3. Okay. So you would want to consider all possibilities. C could be situated in codon position 1. So the next would be position 2, next position 3, 1, 2, 3, 1, 2, 3. There are only 3 positions because codon is made up of 3 nucleotides only. Okay. The other scenario could be that C is situated in codon position 2. So what would be the next position? 3. So 3, and then next would be 1. So cyclic 1, 2, 3, 1. So 2, 3, then 1 will come, 1, 2, 3, 1, 2. Okay. And the third scenario is C is situated in codon position 3. So next is 1, and then 2, 3, 1, 2, 3, and so on. Okay. So considering all these 3 scenarios, 3 possibilities, we can now compute the probability of this sequence to be a gene or to be a protein coding sequence in frame 1. So if frame 1, then you have to consider this. Okay, this is frame 1, codon position 1 for the first nucleotide. Okay. Okay. So let's consider this and compute the probability of this sequence to be protein coding in frame 1 given model order 0. Okay. So model order 0 means probability of C multiplied by probability of G multiplied by probability of G and so on, but now you have to keep track of position also. Okay. So you will put the positions also, which are shown as superscript. Okay. So probability of C position 1, probability of G position 2 and so on. Okay. Similarly, if you have to compute the probability of this sequence to be protein coding in frame 2 for Markov chain model of order 0, so you are considering this. So probability of C in position 2, probability of G in position 3 and so on. Just multiply these probabilities. Okay. And then let's consider the probability of this sequence, frame 1 sequence under Markov chain model of order 2. Okay. Okay. So, and that means that Markov chain model of order 2, two previous bases. So, you are basically considering trinucleotides, right? So, two previous and the next one, trinucleotides. So, and then because of that reason, you have to keep track of the frame. So, yeah, I can, is there something to rub this off? This thing, oh, okay, thanks. Okay, so, okay, let's consider, where did I keep it? Oh, here it is. Okay, so let's consider, yeah, frame 1, this one. So, CGG, okay, so all triplets of nucleotides, okay. So, we consider the model order 2. So, two previous bases and the next base. So, 3 nucleotides. So, CGG and the next would be GGA, and then the next would be GAT. So, GAT, right, and so on. Okay. So, this is, so let's consider the reference position as the codon position for the first nucleotide of the oligonucleotide. Okay. So, this is 1, 2, 3, frame 1, 1, 2, 3, and this is 2, 3, 1, and this one is 3, 1, 2, right? Okay. So, and when we will compute, then we will put the superscript from the reference position that we get from the oligonucleotides. Okay. Okay. So, you did it for non-coding sequence. Okay. You did it for that, and now we are going to do same now for the protein coding sequence, assuming that this is a protein coding. What is the probability of this sequence to be protein coding in frame 1 under Markov chain model of order 2? Okay. Okay. So, same thing. So, probability of starting in CG, and what is the reference position? Probability of starting in CG. So, what is the reference? So, you are considering this oligonucleotide. So, what is the reference position? No, reference position 1. So, that is why put a hat 1 here, that is indicating the position, reference position, and then making a transition to G. So, then probability of G given CG, right? Probability of G given CG, and what is the reference position? 1, right? You are still considering this oligonucleotide, this trinucleotide. So, reference position is 1. Okay. So, reference position 1, and then making a transition to A. So, probability of A given CG. Okay. Probability of A given CG. So, what is the reference position? A given GG, right? Oh, CG. Okay. Did I CGG, probability of starting in CG and then probability of given CG, yes. Okay, I think it is okay. What happened?
Sir, I'm not able to understand the last one, how we position.
Which one?
Frame one model, order two.
Oh, order 2. How we are getting this?
So the positions, one, one.
Oh, 1, 1, 2. Okay, okay. Let's do this again. So yeah, let's consider the sequence, which is CGG. I will write the full sequence: CGG, ATGG, ATGG. Okay. Okay. So Markov chain model of order 2. So basically, we are considering triplets of nucleotides. So 2 previous bases and the next base, right? So, and I am going to consider all oligonucleotides of length 3. So, and this we already know 1, 2, 3, 1, 2, 3, 1. Okay. So, the first one is CG. So, can I get a better yeah, right. Okay, so the first is CGG. Okay, so 1, 2, 3, and I will say that the reference position, because there are 3 positions for this oligonucleotide. I will say that the reference position for this oligonucleotide is the position of the first nucleotide. Okay, so reference position is 1. Okay, good. And the next one is the next, so move by 1 nucleotide. So the next oligonucleotide is GGA. Okay, and this is 2, 3, 1. Okay, so reference position for this is 2. So the next oligonucleotide would be GAT, and then this would be 3, 1, 2, and so on. Okay, okay. So if I have to compute the probability of this with Markov chain model of order, this is a sequence of order 2. So I will write probability of S is equal to, okay, so if I am here, no previous nucleotide, I am here just 1. Okay, so I have to come here to see 2 previous nucleotides. So I will say probability starting in CG. So probability of, I will write first and then I will put the reference positions. Okay, probability of starting in CG and then transitioning to G. So probability of G given CG, and then next is probability of A given GG, and then probability of T given GA, and then probability of G given AT, and finally probability of G given TG. Okay. Okay, so what is the reference position for this? 1. What is the reference position for this? What is the oligonucleotide that this represents? Which oligonucleotide this is? No, no, no. What trinucleotide is this? No, I'm not saying position. I'm saying what trinucleotide is this one? CGG. Okay, so CGG. What is the reference position for this? Now you say 1. Okay, so 1. Okay, what trinucleotide this represents? GGA. So GGA position 2. Position? No, that's fine. Now position 3. So, okay, done. Okay. Okay, so this is how the non-coding sequences and protein coding sequences were modeled using Markov chain models. Okay. Okay, so now once you have the modeling part which is actually computing the probabilities accomplished. Then the other modeling part is that how you can compute the values of these probabilities. You need the values of probability of CG, this is the initial probability, and values of transition probability, probability of transitioning from CG to G, from GG to A, from GA to T, and so on. Okay? So, you need values of these probabilities to compute the probability of the sequence to be protein coding in frame 1, right? And now we have ventured into machine learning. So, you might have heard about machine learning, artificial intelligence, and all those things. So, we are going into that. So, we need to train Markov chain models to learn the values of these parameters. Okay? We want to train a model to learn the parameters of the model, and for that we need training data. And for this example, we need a data set of validated genes, validated protein coding sequences, and another data set of validated non-coding sequences. Okay? So, train a model on validated protein coding sequences and train another model, Markov chain model, on validated non-coding sequences, or 2 models. Okay? So, once you have trained, then you should have the values of these parameters, the initial probabilities and transition probabilities. Okay? And then you can compute the probability of a sequence to be protein coding or non-coding. So, how to estimate the values of the parameters giving training data? So, let us consider simple example. Let us consider the same sequence, and I am going to say that let us consider this a training sequence. Okay? This is a training sequence, and I want to compute the initial probability from, I want to estimate the initial probability or marginal probability of dinucleotide CG from this training data. Okay? So, probability of this dinucleotide from this training data can be estimated from the counts of the dinucleotides. So, how many total dinucleotides are there in this sequence? Okay? All possible dinucleotides. How many total? So, CG, GG, GA, AT, TG, GG. So, how many total? 6. Do it again, 6. You said 5? 6. So, L is the length, so 7 minus 1, 6. So, of 6 of all dinucleotides, so total 6, how many of them are CG? 1. So, the probability of CG is 1 by 6. So, this is how model is being trained. So, model estimated the probability of CG as 1 by 6. Okay? Now, the transition probability of G given CG. Okay? Given CG means 2 previous bases are CG. So, given CG means count of CG in the training sequence. So, count of trinucleotides where first 2 nucleotides are CG followed by any nucleotides. Okay? So, that is basically counting CG in the training sequence. So, of them, how many instances are there that CG is followed by G? No, not now. Not so, I am saying that this is the probability of G given CG. Okay? Given CG, how many of them have G following CG? That proportion is the estimate of probability of G given CG. Okay? Now, you tell me that how many CGs are there in the training sequence? 1. So, denominator is 1. How many CGG are there in the training sequence? 1. So, 1 by 1. So, probability of G given CG is 1. So, the value of this transition probability is 1. So, this is how machine learning models, they learn the parameters. So, given training sequence, they are labeled data training sequences. So, in this example, validated protein coding sequences and validated non-coding sequences. So, train a model on validated protein coding sequences. and then train another model on validated non-coding sequences. So then you will have a trained protein coding model and a trained non-coding model. And then you can compute these probabilities, because now you know the values of these probabilistic parameters. Okay. Okay. So, and this is important, because the question is that whether a given sequence is a gene or not. So, you want to compute the probability of that sequence to be a gene, and you are considering all possibilities. So, the probability of that sequence to be gene in frame 1, then in frame 2, then in frame 3. So, 3 probabilities you are going to compute, okay? And perhaps more when I ask you to go to the next step. So, gene discovery, 3 steps, go back 3 steps, and we did first 2. Okay? We identified features that can discriminate protein coding sequences from non-coding sequences, and then we built statistical models for protein coding sequences and non-coding sequences. The third one we are going to come in a moment, to integrate all these models within a pattern recognition formula or algorithm. Okay? Okay. So, we will come to this in a moment, but I would like you to appreciate the power of modeling in prediction. So, there is a problem in biology which is called CpG island prediction problem. Okay? So, in the human genome, C in dinucleotide CG can be chemically modified by methylation. You understand methylation? Methyl group CH3. So, CH3 can get attached to C cytosine in CG. Okay? And when C is methylated, there is a chance that C can get mutated to T, thymine. And because of this region, CG dinucleotide is rarer in the human genome. However, near the start of the genes, the methylation is suppressed, and because of that region, there is a relatively more abundance of CG near the start of the genes. And when there is an abundance of CG in a region, that region is called CpG island. Okay? So, one of the problems in molecular biology is to identify CpG islands in a genome, and Markov chain models have been used for predicting CpG islands. So, you learnt about how to model these sequences. So, I can skip this because this is for a first order Markov chain model to be used for computing the probability of a sequence. So, I will skip this because you understand. Okay. So, the training has been done. So, the transition probabilities have been estimated, and this is for first order Markov chain model. Okay? So, probability of a sequence depends on the just previous nucleotide or base. Okay? Okay, so let us look at the model of CpG island. So, this is indicated by plus, and this is the model for non-CpG island indicated by minus. Okay? And these are transition probabilities. So, probability of, so this is yes, I think right, probability of A given A. This is probability of C given A, probability of G given A, probability of T given A. Okay? So, all these should sum to 1, because given A you have only 4 possibilities, right? Tossing a coin, you have only 2 possibilities, head or tail, add them up, the probability will be 1. Same thing here. So, rho should sum to 1. So, let us look at the probability of G following C. Okay? So, probability of G given C, which is this one. So, which is 0.274. Okay? So, dinucleotide CG. So, probability of G given C. Okay? That probability is 0.274 for the plus model, for CpG model, And you would expect that for the non-CPG model which was trained on non-CPG sequences, this probability should be substantially less than what you are observing in the CPG model, right? And that is shown here. That probability in the non-CPG sequences is 0.078, substantially less. Okay. So, models have been trained, and now if you are given a sequence, so we will try again. If you are given a sequence, and let us write a sequence and compute the probability of that sequence. So, given a sequence, T, C, G, C, G, A, C, G, G. Okay, this is the sequence. Okay. You want to use these models, already trained models, to predict whether this sequence is a CPG island or not. Okay. So, you would want to compute the probability of this sequence to be a CPG island. So, for that you are going to use which model, plus or minus? Plus. Probability of this sequence to be CPG island. So, you will use plus model. Okay. So, first let us write the probability of this sequence using Markov chain model of order 1. Okay. So, tell me what I should write here. Model order 1, probability of T, then transition probability of C given T, and then probability of G given C, and last probability of G given G. Okay. Okay. And then I will put plus as superscript over P. Okay. And then probability of the sequence to be non-CPG island, so minus, and it would be probability of S to be non-CPG island sequence. So, what should I write? Probability of T. What should I have the hat here? Minus. So, okay, I am putting a hat or superscript, whatever you say, minus, and then probability of C given T minus so on. Okay. Okay. And then I will divide these two, and when I will divide these two, actually I will write equal to here now. When I will divide these two equal to, then what is this called? If I divide the probability of S to be CPG sequence by probability to be non CPG sequence, then what this ratio is often called? Likelihood ratio. Right? Likelihood ratio. Okay. So, likelihood ratio. So, this is the likelihood, and I have divided this. Okay. And when I take the logarithm of this, then this becomes log likelihood ratio, and this is also called odds score. Okay. Okay. So, if this is greater than 1, if this is greater than 1, then what will you infer? The sequence is a CPG sequence or non-CPG sequence? CPG sequence. Okay. And if you are taking logarithm, so if logarithm is greater than 0, if you consider log a score, so if log a score or log likelihood, a score is log likelihood. Okay. So, if you consider a logarithm of likelihood, then this would be greater than, no, likelihood greater than 1. Log 1 is what? 0. So, greater than 0. Okay. Okay. So, which means that if odds score is, so log likelihood score is greater than 0, then this sequence is predicted to be CPG sequence. Okay. Otherwise non-CPG sequence. And the idea is simple, that if this sequence will have tons of CGs, then there would be tons of values 0.274 in the numerator and terms of 0.074 multiplied with each other in the denominator, and this is much higher than this, and because of this region numerator becomes greater than the denominator. So, this is the whole idea. Clear? Okay. So, if you understand this, then I will just skip this complicated notations to describe this model. Okay, you don't have to. If you understand this, done. Don't have to worry about this. Okay, so these models were used for predicting genes. We are coming to the third step, which is to integrate these models within a pattern recognition algorithm that can discriminate protein coding sequences from non-coding sequences. Okay. So, given a sequence, and this is what we already discussed. So, given a sequence, the probability of that sequence to be protein coding in frame 1, frame 2 and frame 3 can be computed, something like this. We just did it, and this is for a first order Markov chain model. And the probability of the sequence to be non-coding can also be computed. Okay. This is homogeneous Markov chain model description, and these are non-homogeneous Markov chain model description. Okay. Okay. So, once you have computed these probabilities, then you can plot them. Okay. And what researchers did actually earlier, that they moved a window over a genome and then computed the probability of the sequence within the window to be protein coding in one of the 3 reading frames to be the non-coding. So, here is the genome, and a window was moved, and window was of length 96 nucleotides. Okay. Window was moved and then slid every 12 nucleotides. Okay. So, next, and then next it was moved, and the probability of the sequence within the window to be protein coding in one of these 3 reading frames. So, frame 1 and then frame 2 and then frame 3, and then the probability to be non-coding were computed. Okay. And then they were plotted actually. So, okay, so computed, and then these were integrated within a pattern recognition algorithm. Okay. That was the third step. So, compute this and integrate all these models within a pattern recognition algorithm, and a popular such integrative model is Bayesian formalism. Have you heard about Bayes theorem? No? Okay. Oh, you have heard? Okay, good. Yeah. Bayes theorem is probability of x given y is probability of y given x, probability of x, and then sum over x, probability of y given x. Can I get a better mark? So, all these are drying up because you can't see from there, right? So, okay, I will show you that, don't worry. I have put all these things on the slide. So, probability of Bayes theorem, probability of x given y is equal to probability of y given x multiplied by probability of x divided by probability of y given x multiplied by probability of x and sum over all x. Okay, Bayes theorem. So, what it does, it computes the posterior probability of x given y. Okay. So, let us say that, and again example with the DNA sequence, let us say that I have a sequence, and we know that bacterial genomes are packed with coding sequences, right. 90%, about 90% of a bacterial genome is coding, only about 10% is non-coding, right. So, I will ask you to predict. I have a sequence, and I have not shown you the sequence. I will ask you to predict whether the sequence is protein coding or non-coding, and you will be tempted to predict coding because 90% of the, I will tell you that it is bacterial. You will say that, okay, I can win if I will say coding because there is a 90% chance of winning, right. So you would be tempted to say protein coding, because in bacterial genome 90% of the genome is protein coding, 10% only non-coding. Okay. Now I will show you the sequence. I will give you the sequence. So your earlier belief, actually 90%, may now change given the evidence provided to you. Okay. So the probability before seeing the evidence is called prior probability, Px. Okay. This is prior probability. Why is evidence? Thank you. Why is the evidence? Why is the sequence given to you? Why is the evidence? So once the evidence is given, so probability of, so X is sequence. So X is not sequence, Y is sequence. So Y is the evidence sequence. Okay. X is model actually, and you said protein coding, so that's protein coding model. Okay. Okay. So probability of protein coding, you said, okay, you are tempted to say 90% because of your belief, because of your prior knowledge about the bacterial genomes, you will say 90%. Now the evidence is given to you, which is sequence. So your belief will change, and this is called posterior probability. Probability of model or probability of protein coding given the evidence, which is the sequence. Now your belief will change after having seen the evidence. That is why this is called posterior probability. Okay. Posterior means after having seen the evidence. Okay. So this will change, and importantly, if you see the denominator, X is model. So all models can get integrated through Bayesian theorem, right? In the denominator, it is summation over X, and I said X is model. Okay. So how many models are here total? 1, 2, 3, 4. Okay. So all these can be integrated within the Bayesian formalism. Okay. Bayesian theorem, which is in the denominator. So let's say that X is model and Y is sequence. Okay. So then it would be probability of a model given the sequence, given the evidence, is equal to probability of sequence given model multiplied by probability of model, and sum over all models, probability of sequence given model, probability of model. Okay. And so here it is. So let's consider coding model. So let's consider this one, coding model frame 1, and I would say frame I, where I is 1, 2 or 3. Okay. So posterior probability of protein coding, so protein coding in ith frame given this sequence, okay, is equal to, use this one is equal to probability of this sequence given the coding model in frame I multiplied by coding model in frame I. So same thing actually, use this, and then sum over all the models, the 3 coding models and the 3 and the non-coding model. Okay. Do this. Okay. So this will give you the posterior probability of the sequence to be protein coding in frame I, where I goes from 1 to 3, right, 3 frames. And then you can also compute the posterior probability of the sequence to be non-coding. And when you sum all these 4 probabilities, they should sum to 1. Okay. Just see, just add these up. So these are 3. I goes from 1 to 3. So first you would compute probability of coding subscript 1 given S. Okay. I is equal to 1. Then you would compute probability of coding subscript 2 given S. Okay. And then you would compute probability of posterior probability of coding subscript 3 given S. Okay. And finally you have probability of posterior probability of non-coding given as total 4. Add them up, you will see that it is 1. Okay. Numerator will be same as the denominator. Okay. Where? Oh, J is indicating that this J goes from 1 to 3. Okay. So this is summation. So first consider J is equal to 1. Okay. So, j is equal to 1, then this becomes probability of S coding 1. Okay. Probability of S given j is 1 coding in frame 1. Okay. And then multiplied by probability of coding in frame 1. So, and then next is, so this is, so yeah, so this summation is from here to here because there is no j here. Okay. And then next is j is equal to 2, and this summation means that keep adding up as you move to next j. Okay. So, add to this which I just mentioned, j is equal to 2. So, when you consider j is equal to 2, then this would be probability of S given coding subscript 2, which means frame 2, okay, multiplied by probability of coding subscript 2, okay, and then plus, now j is equal to, consider j is equal to 3, okay, plus probability of S given coding, given code subscript 3 multiplied by p coding subscript 3, okay, and so done all three. So, goes from j to 3, and then you add to that the probability of S given non coding multiplied by probability of non coding. Okay. So, total would be how many terms? 3. So, in the denominator of this. So, when you expand this, when you are just adding, so you will have 3 terms and this would be the 4th. Add them up, this is the denominator, right? Clear? Okay. So, yeah, unfortunately, I am not able to, because of time, I am not able to do everything on board. But you would try to understand and ask question, otherwise I do not know whether you are understanding or not. It is just not possible. These are mathematical models, but I want you to appreciate, without this omics course is incomplete. Okay. Omics is not just use of software tools or web servers. Omics goes deeper into mathematical modeling and development of algorithms. Okay. 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 So, once these probabilities were computed, posterior probabilities, then they were plotted. So, a window was moved over a genome, and for each sequence in the genome, these probabilities were computed, and then those probabilities were plotted. So, this is frame 1. Okay, so frame 1, and then next 2, frame 2, frame 3. Okay, let us consider this only. So, probabilities were plotted. So, probability go from 0 to 1. Okay. So, they were moved, and then at some position. So, this is also genome position. Okay. On x axis is genome position. Okay. So, at some position, I would say that at this position there is a gene, and that gene is located in frame 1. So, the probability of this window to be protein coding in frame 1 will be low or high. I say that at this location there is a gene. So, the probability of that window that contains this sequence at this location to be protein coding in frame 1 will be high or low? High, because here is a gene. So, let us say that that position is this. So, suddenly there would be a spike here, probability, okay? And then gene is several windows, right? Gene could be, this is a window of just 96 nucleotides, gene could be 1000 nucleotides. So, for several of these overlapping windows it will remain high, and then it will drop, okay? So, and then you will say that, looking at this profile, you will say that, okay, there is a gene at this location in the genome, and that gene is situated in frame 1. Okay. So, very good. So, this is what was done. Yeah, try, please try, otherwise, yeah, I will not be able to cover many of the things. I am trying to help you understand with as simple descriptions as I can make, but these are not as simple models. They have been used for annotating genomes. So you can see that at this location, from this to this, from here, this position to this position, there is a gene, and that gene is in frame 1 on direct strand of DNA molecule. Okay? So there are 3 frames for direct strand and 3 frames for complementary strand, and 1 more frame would be for non-coding. Okay? Okay. So what earlier people used to do, they were analyzing DNA strands in turn. So first direct strand and then complementary strand. Okay? So when you do for the direct strand, then you are computing these probabilities. So 3 here, I goes from 1 to 3 and non-coding. Okay? And then you do for the sequence in the complementary strand. Okay? So do in turn. So when this was done, so first the probabilities were, posterior probabilities were plotted for the direct sequence and then for the complementary sequence. And what was observed, that the strand opposite to where a gene was earlier predicted, that location also had some signals. Okay? So a gene was predicted here on the direct strand at this location. Opposite to this is a region on the complementary strand. Okay? And there is no, actually there is no gene there, but signals were observed, and that led to many false predictions, spurious predictions. So there is a gene, and people said that, let's say, gene shadow. So there is a gene in this strand and there is another strand here. So on the other strand, opposite to the gene, there was a prediction. Okay? So in the gene shadow region, there was a prediction because of these spurious signals that arose. And people started thinking, why these signals are coming in the region opposite to, in the region of the strand opposite to where the gene is located. Okay? Why that is happening? So finally they were able to figure this out. They found, as I said, that there is an abundance of R and Y codons, okay, in genes, in coding sequences. And let's complement this. So this is on the direct strand 1, 2, 3. Okay, so 5 prime to 3 prime will read like this. Okay, 1, 2, 3 is these are the codon positions, and on the complementary strand I would read in the opposite direction, 5 prime to 3 prime, but from the other direction. Okay, so what is the complement of this? So this is pyrimidine. Pyrimidine is C or T. So what would be the complement of C or T? So C G and T A, right? G A, and N is any nucleotide, so N N, and this is purine. Purine is A or G. So what is the complement of this? C or T, right? So this C T is Y, and this is N, and this is R, and this is 5 prime, and this is 3 prime. Okay. I think I should throw these, otherwise it's confusing actually which one. Okay. So now you have to read in this direction. So then you will read R and Y. Okay. So on the other strand, there was again an abundance of R and Y codons. Okay. So on the strand where there was a gene, of course there was an abundance of R and Y codons. On the other strand opposite to where the gene is located, there also in the gene shadow region on the other strand there was an abundance of R and Y codons, because R and Y codons are self complementary. Got it? Complement and read from the other direction. That's again R and Y. So because of this pattern, there was a false signal on the other strand. So you can see the false signal. And this also happened because researchers were analyzing these strands in turn. So, when the model has seen this, model has not seen this, or when the model has seen this, the model has not seen this. This is a very strong signal, and this is relatively a weak signal compared to this. Okay? But because model has not seen this, and if you remember, if you sum all these up, sum should be equal to 1. So, model, for example, for the shadow region, model gets most of the strength from here and rest are almost 0. Okay? And that is why this gets most of the strength is close to probability 1, right, at most of the locations. But if you allow model to see both the strengths together at the same time, then it is possible that this signal will pale in comparison to this signal, because there would be 3 for this strand, and then 3 for the other strand, and then non-coding, right. So, when the model has seen all, then and they all should sum to 1. So, this gains most of the strength, because this is so strong, this will be close to 1, this will get to 0 for that region, right. So, you have to show the model everything at the same time, and this is the beauty of machine learning. So, do not do in pieces, let the model see everything, so that model can take right decision, just as human can take a right decision when human is shown all the evidence at the same time, not in pieces. Okay? Yes. What? हो सकता है gene, but in bacterial genomes, genes do not overlap like this, they overlap at the ends. So, this is well known, and that is why people wondered that why this is so. So, in the gene shadow of every gene, there was this signal, and that is a false signal, because in bacterial genomes, genes do not overlap fully, they do not overlap like this, they can overlap at the ends. Okay?
But in viruses, it says the gene overlap like this.
Which one?
For viruses.
Viruses can overlap fully. Okay, then that's another thing, but yeah, but here they were focused on the bacterial genomes, and in bacterial genomes, 2 genes do not, on the 2 strands, they do not fully overlap, they overlap at the ends. And this is something that has always been incorporated in the prediction models of bacterial or prokaryotic genomes. Often models assume that 2 genes do not overlap at all, earlier models. So, there is at least one nucleotide between 2 genes that are on different strands. But now later models, they also included the modules for overlaps to predict overlapping regions in the genes. Okay. So, if model has seen everything, then the model should be able to predict correctly the gene, right. It would be ordered to eliminate the spurious signals. So, this is a spurious signals, it will just get discarded, because this is so weak compared to this. Earlier model was not shown this when model was computing this, and this was gaining most of the strength, sum should be equal to 1. So, it showed like this compared with other weak signals, but when model is compared with this strong signal at the same time, it gains most of the strength, is close to 1, this becomes 0. Okay? Yes. Yeah, this is for only prokaryotes. Eukaryotic model would be different from this, even though the idea will remain same, that you have to build a model for protein coding genes, and then so you will say model for exons, the coding sequences, and then a model for non-coding sequences, and model for introns or splice sites, and so on. So, you will have more features that you have to consider for eukaryotic gene than for prokaryotic gene. So, to help you understand this, I have considered the simplest model, that for the prokaryotic gene. to help you understand how models are used for prediction. Okay, so I think we discussed this. So now how many models will be there? 3 models for coding, 3 models for gene shadow of gene, right? Shadow of gene. So how many? 6, and 1 for non-coding, total 7. So total 7 models. So when 7 models were used, then of course you compute the posterior probabilities of these 7. So probability of sequence to be coding in one of the 3 frames, probability of the sequence to be shadow, gene shadow, again in one of the 3 frames, and non-coding. Okay. So when that was done, can you see the signal is false signal is gone? It's gone. Okay, and this program was named GeneMark, and was used in annotating the first bacterial genome that was sequenced in 1995. Do you remember the first bacterial genome that was sequenced? No, not E. coli. E. coli 1997. First bacterial genome sequenced in 1995. E. coli maybe second, not first. No, not that. Not that. Have you heard about Haemophilus influenzae? Okay. It does not cause influenza. That's a virus actually. H. influenzae, Haemophilus influenzae. It's a shorter genome. I think 1.5 megabases or 1.7. You can check actually. So around that. So this program was used for finding genes in that genome. Okay, so and here is a posterior probability profile generated by GeneMark program. Actually, you can go to you can just Google search GeneMark. You can go there and you can copy paste a genome sequence, and it will provide you where the genes are located. Okay, so now that you understand, you should be able to. So let's look at the profile generated by GeneMark. So can you let me know how many genes are there in this sequence, which is starting at this position? Okay, let's say 0th position. Okay, from this position to this one, which is about 20,000 something. Okay, whatever it is. Yeah, 20,000 something. Yeah. Okay, that's fine. So how many genes are there in this sequence? Is this a gene? Looks like, right? Yeah, looks like this is a gene. So and they put a threshold here that the windows should have the posterior probability greater than 0.5 and many of them, right? So eventually they take average and then they ask whether it is better than or greater than the threshold or not. Okay. So there is a gene in which a strand, direct strand, okay, in frame 1. Very good. Now you understand. Yes. And these are noises. Okay. Again, this is a noise, it seems like. So second gene is in which strand? Complementary strand. Which frame? First frame. And then third gene is here. Which frame? Third frame. And fourth gene also in third frame. So thus you know actually where the genes are. So this is the output that program will provide you. Okay, run GeneMark, it will give you this, and then you can go through this profile and you can say that okay, there are 4 genes and they are located from this position to this position and they are in these frames. Okay. Now when you run GeneMark, then you should be able to interpret the output from GeneMark. Okay. Okay. So one of the challenges with the use of Markov chain model for predicting genes by GeneMark was that the predictions were made for windows. Okay. So the probabilities were computed for the sequences within the windows, and which means that the model has not seen the entirety of the information that is there in the entire sequence. It is seeing only the information that is contained within the window, and that is why they did heuristic. They tried many different window sizes. So, longer window sizes, good, more information, but it reduces the resolution of finding the boundary between protein coding and non-coding sequences. Okay? If you increase, because the resolution is as good as the window size. If you as good means that, okay, if it is longer, then resolution is bad. Okay? If it is short, then you are getting close to the location where the boundary is between protein coding and non-coding. Okay? As short, if it is just one nucleotide resolution, that is the boundary, because boundary is just one nucleotide. Okay? If it is longer, then resolution gets worse. So, that was a problem, and the other problem was that when you move a window, and let us say that this is the boundary, okay, and window is here to here, and this is the boundary. So, a part of the window is protein coding and another part is non-coding, right? But the entirety of the window has to be predicted protein coding or non-coding. So, there is another limitation. That a window that is spanning the boundary of protein coding and non-coding has both protein coding and non-coding parts, but the model is constrained to predict the entirety of the window to be protein coding and non-coding, right. So, that is a problem. So, to address that problem, people started thinking about new models, and one of the models, again based on Markov chain model, is called hidden Markov model. So, hidden Markov model can consider the entirety of the information that is within a given sequence for predicting a nucleotide to be protein coding or non-coding, which means that the resolution is at the single nucleotide level, right, no window, entire sequence information, and you predict based on this entire information a nucleotide to be protein coding or non-coding. Okay? And because of that reason, the other limitation also gets addressed, where a window is spanning boundary between protein coding and non-coding, and entirety of the window has to be predicted either protein coding or non-coding. But with this model, hidden Markov model, you do not have to worry about this, because each base is now being predicted as protein coding or non-coding using the information that is there in the entirety of the sequence. Okay? So, then you would wonder what is the hidden Markov model, and it has been frequently used in the field of omics, proteomics, transcriptomics, genomics, to name any. Yes. T, okay, I am not keeping track of the time. Okay, so let's have a tea break and we will come back. Okay. We will be back around 12. Okay. So, yeah, tea break, we should come here around 12. Okay. We will continue, and then we will have lunch break, and we will come back around 2. Okay. So, let us use the time so that you learn modeling today and do some tutorials also. Okay. Okay.